Very, very tantalizing. Yeah, I remember the moment these images came down. We were actually had a rover science team meeting. I think it was the next week, or maybe it was later that week. And what an exciting time to be on the team looking at this data because we pulled those images and it's, oh my God, this is so remarkable. We have it's been the objective of our mission to find these potential biosignatures, and there's many people on the team who've dedicated their careers
Yes.
to finding these. So when we finally saw these images, well, it was incredible.
What's up, you scholars of enlightenment? Dr. Sam Gregson, particle physicist here again. I hope you've had a brilliant weekend and are enjoying a productive start to the week. Now, a couple of weeks ago, NASA made a fascinating announcement regarding a potential biosignature on Mars. The Perseverance Martian rover has been exploring the Jezero crater region on Mars, the site of an ancient Martian river delta, since 2021, and has been assiduously searching for evidence that life may have once flourished on the red planet. In 2024, Perseverance took samples from mudstone and conglomerate outcrops known as the Bright Angel Formation on the edge of an ancient river valley known as Neretva Vallis. Perseverance drilled into the rocks at the formation and analysed samples of their interiors. The rover revealed something astonishing: nodules and reaction fronts enriched in organic chemicals and iron-rich compounds. On Earth, such nodules are associated with microbes interacting with iron-rich environments, such as peat bogs. Astrobiologists were understandably incredibly excited. So have we found solid evidence of life on our nearby planetary neighbour? Or are we jumping the gun? To help me answer that question, I'm joined by a brilliant special guest, Mars scientist Henry Manelski. Manelski is currently a member of the Perseverance rover SuperCam science team. The SuperCam on the Perseverance rover examines rocks and soils with a camera, laser and spectrometers to examine the chemical composition of the Martian surface and explores the Martian geochemistry using LIBS and visor spectroscopy. His research has focused on comparing LIBS and visual reflectance spectroscopy on Mars, using LIBS plasma diagnostics to explore the fundamental assumptions of SuperCam's elemental calibration and investigating trace metal enrichments in Jezero crater. Manelski is a scientist who studies the chemistry and history of Mars every day. So who better to help me understand whether we really have found telltale signs of life on the red planet? So, Henry, before we get into the latest results from Perseverance, can you just remind us, what is the rover's mission on Mars and why are we particularly interested in the Jezero crater? If I get this a little bit more centered on our screen here, why are we roaming around Jezero?
Great question. Well, I think most people are probably aware a lot of our exploration of Mars, NASA's exploration of Mars, has been driven by looking for life. The search for life is our ultimate goal. And a big part of that is the search for water, right? Life as we know it on the surface of the Earth requires water. Water is a fantastic solvent for life. So we've been trying to explore places on Mars that we think we have a good chance of finding rocks that were shaped by water, chemically, morphologically, and therefore might hide evidence, maybe fossilized evidence of ancient life on Mars. So the reason why we are so interested in Jezero crater is Jezero is like a 45-kilometer impact crater. You have a great artist rendition here on the screen showing of what it probably looked like 3.8 plus billion years ago. This impact crater was once filled with water. There was a lake there. So on the western side, which we've been exploring, there's this deltaic feature, this fan, much like you might see in the Mississippi River Delta or something. This is a feature that formed when water moving in from a river with a whole bunch of sediment and rocks mixed up in it met a standing body of water, a lake that was in Jezero crater, and then it deposited all of those sediments, forming this weird deltaic feature with these lobes. So this is really our big evidence that we had from orbit that told us there was a standing body of liquid water here, and that's probably the most compelling reason why we went to Jezero. A shallow lake, these are the kind of environments that on Earth are, really good for the preservation of life. So yeah, that's why we went there.
Fantastic. So, what we are told recently, or the recent news that came out, was that the rover had potentially found a biosignature in Jezero crater. So, when we talk about a biosignature, what are we talking about here?
Great question. Yeah, I think they're very careful about the wording. They, rather than potentially finding a biosignature, I think how we described it was we found a potential biosignature.
Yes, yes.
Uh, and
This is how
biosignature,
scientists talk, right? It's very, very
yes,
careful with what we're actually claiming.
nomenclature is pretty specific. When we say biosignature, what we mean is something that could have been formed via processes that involve life,
Yeah.
right? Often this idea is wrapped up in the way we look for life in ancient sediments on Earth. So the search for ancient life on Mars is really exciting. And in many ways, it's paralleled by a search for the most ancient life on Earth. There are really old rocks on Earth that we're looking for life in, and we're often looking for what we'll call biosignatures, which are morphological features in the rock, chemical features in the rock that we believe we can definitively tie to biotic processes. Really ancient rocks, that means microbes,
Yeah.
single-celled organisms, the types of things that the structures they might form in the process of their metabolism. In the case of fossils, it's the structures themselves, of those cells. So when we refer to a biosignature, we're referring to evidence of chemicals, morphologies, so little features that you could see in the rock that could have been created by living processes. But it's a really loaded term, of course, and we'll get into this in this interview, I'm sure.
Absolutely. I seem to remember when Perseverance was first going up, some of the initial runs on the Earth that were all about could they identify these cyanobacteria structures, for example, in rocks on Earth, and would we be able to find similar structures on Mars? So you mentioned that those parallels there. It's not enough, right, just to find biological molecules, right? We also have to have that other part where they're not, where it has to be produced by living processes, right? It's not enough just to find particular organic molecules, right? Because we found organic molecules, for example, in star forming regions. We find them in, plumes coming off, moons in the solar system. We find Curiosity found organic material, for example, on Mars previously. So you have to have those two parts, right? You have to have the organic materials which life could produce, and also evidence that biological processes themselves produce those molecules. Is that fair? You have to have those two parts linked together.
Absolutely, especially in the context that we're going to be talking about organic materials today. We're going to be talking about the detection of G-band organics, which is just carbon-carbon bonds.
Right.
So there are many ways that could have possibly formed. We have to be able to definitively tie that to biotic processes. Of course, yeah, you mentioned organics found in star-forming regions, organics forming in asteroids, airless bodies. So it's places all over the solar system. It's about the complexity of the organics too.
Yeah.
So the more and more complex the organic molecules are, the longer the carbon chains are, the more likely that life was involved. So things like fats, like lipids, that kind of thing, you probably more associate with life. But amino acids, which of course are essential to life, are found in chondrites. And that is well known and not necessarily tied to biology.
Seem to remember
So you need
the Miller-Urey experiments, creating them in flasks with all sorts of simple molecules and a little bit of electricity. You can get these molecules quite developed molecules, quite intricate molecules, just by abiotic processes.
Yep, certainly. You need many lines of evidence, extraordinary claims, extraordinary evidence. Yeah.
Absolutely. So, what samples did Perseverance take that facilitated the recent analysis and the claim of a potential biosignature? So, Perseverance has been roaming around Jezero crater. Which samples that it's been taking are we talking about for the recent results, which have got so much media fanfare?
So we mentioned why we went to Jezero Crater and that delta that we found in Jezero Crater.
Yeah.
So we went over the delta and then we went into that river channel, which once
Yes.
brought water into Jezero Crater and built out that fan. That is Nuret Vivalis, which you can see here.
Mm.
So going left to right is that river channel. Water once moved from the left to the right of your screen. That is what we explored. When we went into Nuret Vivalis, we found these two bright outcrops that you can see from orbit here that are labeled Bright Angel in the north, Masonic Temple in the south. These are mudstones. So really fine-grained sedimentary rocks that were probably formed in a low energy aqueous environment like a lake. We did a whole lot of exploration over this entire region. You can see in those triangles there, those are pixel scans where we got X-ray fluorescence observations, elemental maps, which are really critical to the story. In terms of the actual samples that we took, samples that could be returned to Earth
Okay.
as part of Mars sample return, there's only one.
Right.
There's the sample that we took in the Chava Falls, Apollo Temple, Steamboat Mountain workspace that's called Sapphire Canyon.
Right.
So we only took one sample, but everything else you see here are those pixel scans, which are really important to the story, right? Not only did we take a sample that hopefully one day will be returned to Earth and we can do all these analysis on, but we had a tremendous amount of context that we got with our rover instruments. That's crucial. Yeah.
So just to be clear for people watching here, there was only one drilled sample that was then stored for later return, which is on this Jezero Falls rock, which we call the Sapphire Canyon sample. I think I've got a little image here. So there was a drilled sample on the Sapphire Canyon. Otherwise, samples were not taken, but scans were made of these rocks in the context that they were in, and we can get information based on those scans. So we're gonna come on to that now. So what did Perseverance find about these rocks or in these rocks that has scientists so excited about the discovery of, again, a potential biosignature on Mars? What were the exciting findings from these rocks at these two locations, Bright Angel and Masonic Temple?
There was a lot of separate lines of evidence that came together to make this particularly compelling. Probably just the first order thing, the background is this is a mudstone.
Yeah.
We know mudstones are probably the best in terms of preserving organic molecules, preserving the morphologies of potential biosignatures, and also just as a habitat for life. Mudstones on Earth, when they're a sediment, fine-grained sediments in a lake, are great for life. Life loves to hang out there. Contrast that with like an igneous rock, right? We don't want to be looking for biosignatures in something that was once in a volcano.
Yeah.
So that's the first order thing. The things that really made this tantalizing, of course, we took pixel scans. So pixel is an X-ray fluorescence instrument. So it creates these centimeter scale maps. Pixels on the arm of the Perseverance rover creates these centimeter scale maps of various elements that are present in the sample. And we also, this is great. You can see a visible image here. And what we found are these spots of locally reduced sulfur and iron. That is that bright material. You can see that in the visible images too, which is reflected in this pixel scan, surrounded by these dark rims of iron phosphate, vivianite. And this looks a lot like a reduction zone or reduction feature that we see in ancient sedimentary rocks on Earth. In the Archean, around this time, 3.5 plus billion years ago. And we think these morphologies, these leopard spots, as we call them, those big ones, formed from life on Earth in these ancient sedimentary rocks on Earth. And we'll talk more about how they form on Earth because there's some important nuances there.
But the initial, so just going through the story so far. So we've pulled up at a place where we believe it would be likely that life would have developed, would have hung out, would have been trapped, essentially going forward.
Would have been preserved.
Would have been preserved, exactly right. And then we've looked inside, we've looked at these rocks, and we see features that look like the kind of things life would produce on Earth. So this is very, very indicative, potentially, of life. And now we're going to go into some more of the analysis. But this is very immediately very exciting.
Very, very tantalizing. Yeah, I remember the moment these images came down. We were actually had a rover science team meeting. I think it was the next week, or maybe it was later that week. And what an exciting time to be on the team looking at this data, because we pulled those images and it's, oh my God, this is so remarkable. We have it's been the objective of our mission to find these potential biosignatures, and there's many people on the team who've dedicated their careers
Yes.
to finding these. So when we finally saw these images, oh, it was incredible. The thing that really made this compelling as well is we had these morphological features which we can relate to life with chemistry that looked similar to features produced by life in ancient rocks on Earth.
Yeah.
But then we also detected organics.
Yes.
And this is the first time we had done this. So using the Sherlock instrument, which is a Raman spectrometer, which is also on the arm, just like the Pixel instrument, we did scans of this same area. And with the Sherlock instrument, we detected a G band. You can see that there on the right. G stands for graphite or graphene. This is carbon-carbon bonds, the kind of bonds that you find and the kind of spectroscopic signature that you find in disorganized organic matter on Earth. So in this context, in a mudstone, you might often find this from kerogen, sort of like fossilized plant material and biomass,
right.
Which is, of course, really, really exciting. The fact that we had both of these lines of evidence together, and the fact that as soon as we left this area with these macroscopic biosignatures, that G-band signature went away. So it wasn't a fluke, right? It wasn't that this was something weird with our calibration.
Yes.
Both of these lines of evidence appeared together, and then they went away when we left.
So we now have a context where we'd expect life to be preserved. We have an observation of the kind of chemicals that could be produced by microbes or are produced by microbes in these kind of environments on Earth. And we have a G-band detection of organic material, carbon-carbon bonds. So we've got all three of these things kind of coming together as lines of evidence in this one spot and not in other spots.
Yeah, this is a particularly reducing area. You can see how those white spots are mainly reduced iron. They're not ferric iron, which mostly looks red. That's why Mars looks red. There's a lot of ferric iron. As soon as we go to places that are more oxidizing, like just south of Bright Angel in Masonic Temple, where most of the rocks are really red, they're oxidized, the kind of environment where you would expect organic matter to break down and oxidize, we don't see this G-band signature. So that seems like a pretty compelling story from our point of view.
So is the story here perhaps just to be clear, because it's potential that I misunderstood this. So it seems to be the case that the idea is that these microbes, if they exist on Mars, are kind of taking the ferric iron that you mentioned, this Fe3, this is the iron that makes the surface red on the red planet. It's rust, essentially. And they're reducing this down to the Fe2, which is the whiter form of iron, this ferrous iron, this Fe2 that we see. And where we see more of this reduction happening, where we see more of this potential biological activity happening, as we see on Earth, we are seeing a stronger G-band signal at the same time with more carbon-carbon bonds. So this is suggestive that where there's more of this organic material, there's more of this on-Earth microbe-like reduction happening. So we have this kind of correlation between the amount of reduction and the amount of potential organic material that's there. And that is suggestive of the idea that more microbes, more reduction. Is this kind of the idea we can see in this plot? As we get a stronger G-band, more organic material, we seem to see more of this signature.
Yes, I think so. That's one angle.
Okay.
There's a lot of microbes on Earth that reduce iron, chemosynthesis. This is how they get energy. That's certainly a possibility here.
Okay.
I think the angle that I'm more familiar with from this work, this could be just part of my biases as a scientist, is sulfate reduction.
Right.
Uh, Iron reduction it can be challenging, but sulfate reduction is really difficult. To turn sulfate into sulfide
Mm.
on Earth, we'll talk about this a little bit later, you really require things to be very hot,
Mm.
or you need microbes. In low energy conditions, the only way that we know to reduce sulfate to sulfide, which is what we see in those light tone areas, is with microbes.
Right.
So on Earth, in really ancient sediments, in the Archean, long time ago, billions of years ago, pyrite, iron sulfide, fool's gold, was a pretty significant fraction of the sedimentary rocks on Earth. Now it's not because it rusts so
Mm.
easily. But before the atmosphere had a whole bunch of oxygen in it, pyrite was all over the place. And it formed via microbes in the water column, in a context like a lake, via what's called sulfate reducing bacteria, where they took sulfur that was in the water column, sulfate ions, and reduced that
Mm.
to native sulfur, hydrogen sulfide, and this produced iron sulfides with iron that was just around, present in the water column. So in that case, it's more that they are reducing sulfate to
Mm.
sulfide. That is really challenging, and I
Right.
think that's
The more
the
compelling
more
signal
compelling.
than the iron signal.
I think
Interesting.
so. But both of those are certainly possible. There are plenty of microbes on Earth that use iron reduction as part of their metabolism, and that could very well be what we're seeing. It could be a combination of the two,
Mm.
for example.
Amazing. Very, very, very interesting. So we've got all of these lines of evidence potentially pointing towards the possibility that we have life doing its thing here on the red planet. So why is all of this stuff? So we talked about all these lines of evidence. We've got the context. This is mudstone. We'd expect it to preserve life. We have these this reduction of sulfite to sulfide. We have potential reduction of iron. We have these in a context where we would expect bacteria to do things like this on Earth. Why is this not a slam dunk? Why is finding all of this and all these lines of evidence not a definitive discovery of a biosignature? Why are we still saying in our paper, you know, redux driven mineral and organic associations in Jezero crater Mars? Why are we not saying, hey, life found on Mars, let's go?
Very good question. We'll talk about this a little bit later, I think, but
Sure.
in the past, we've jumped to conclusions too quickly, and we
Yes.
want to be careful. That's probably the main reason. But there are ways that this could potentially be explained without biotic processes.
Yes.
The main one, in the case of sulfate reduction, it's high temperature.
Mm.
If this rock was ever exposed to conditions over about 120 degrees Celsius, you could potentially produce reduced sulfur and these pyrite nodules that we see. You could potentially produce them without any biology required.
So we have a chemical pathway that we know about from chemistry on Earth where we could do this with an abiotic process, and therefore there's
Yes.
another production mechanism that we need to rule out,
Mhm.
at least one
And
to rule out.
yeah, there's reasons why this maybe seems unlikely that it was heated to this extent. So this is a mudstone. We investigated this mudstone a lot. One of the things that was pretty clear from right when we arrived is this rock is barely a rock. It's really friable. It's really delicate.
Right.
Like when we drill into it, it just fell apart basically,
Yeah.
which tells us to first order, it probably wasn't buried a whole lot, which is what is required to heat it up. It needs to be buried quite a bit. So we think it probably formed close to the surface. It
Yeah.
probably wasn't buried a whole lot. And if it was heated a whole lot, you might also expect these tiny little mudstone grains to have recrystallized into
Yes.
bigger features, which we
Yeah.
could then identify.
Yeah.
So it doesn't look like it's been heated. But that possibility remains. And because that possibility remains, we should be careful.
So this is kind of
Um,
like, would this be kind of like on the Earth where, granite potentially cools slowly under the ground? We see these
mm,
large crystals in it. We don't see that in
mm,
this rock. So that's potentially some
mm.
evidence it wasn't heated and pressurized to a significant degree in the past. And that gives us some confidence that this high temperature abiotic process maybe wasn't what made these leopard spots and poppy seeds. Is that a fair layman's kind of
Yeah,
assessment?
certainly. Yeah. And then beyond that, there's this just whole category of this is another planet.
Yeah.
There could be chemistry that we don't
Yes.
quite understand. These are the two ways that we know of reducing sulfate on Earth,
Yeah.
thermochemical sulfate reduction when things get really hot, or bacterial sulfate reduction.
Yes.
But there could be a third one that
Yeah.
we haven't encountered yet. This is a whole new planet. So I think that's another reason why we're describing this as a potential biosignature and trying to, we need to cross our T's, dot our
Yeah.
I's, and really be certain of this.
And you've teed that up really nice. It's a very nice segue because it must be, I've said is it, but it must be very difficult to prove the operation of biological processes in an entirely alien environment. You're but essentially trying to prove the existence of life almost by elimination of these processes. But
Mhm.
of course, do you know you've got all of them on Earth for a start, and then
Mm.
we're kind of translating onto a completely alien environment, aren't we? So is it very difficult to do that kind of process of elimination? Because I've seen another couple of these where, for example, the potential discovery of a biosignature on K218b, very
Mhm.
difficult to rule out this dimethyl sulfide was potentially made by life or by an abiotic process. This is a completely alien, different kind of world, very different even to Mars. We can think about the phosphine detection even on Venus, which is had
Mhm.
an awful lot of contention around it. Could we make phosphine by an abiotic process in the clouds of Venus? Can we not? Are there bacteria in the clouds of Venus? Are there not? It seems very difficult to do this kind of process of elimination, right?
Mhm. Yeah, the process of elimination is difficult, requires a tremendous amount of context. We don't know the average surface temperature of Mars at the time these rocks were formed. In fact, we don't even know when these rocks were formed, right? The timing of Jezero crater, even for example, the crater counting, which is generally how we date planetary surfaces, is has huge error bars on it.
That's
So
right.
we don't really know when these rocks formed, uh, and we don't know what the chemistry of the atmosphere was like. There's so many big question marks that we have for Mars. Maybe if we start nailing that down a bit better,
Yeah.
we can start to rule more potentials out.
Yeah.
Um, which is why we really tried to thoroughly document the context in which we found these biosignatures. We spent a long time there, taking a lot of these pixel scans, taking a lot of images, so that hopefully when the samples maybe come back one day,
Yeah.
we'll have a lot of reference material. That's the idea.
Sounds like the best way to go. So how strong would you say this kind of claim is at the moment? We've said that the scientists are obviously very careful not to get out over their skis. We have the lead author, Joel Horowitz, of this study saying that the combination of chemical compounds we found at Bright Angel could have been a rich source of energy for microbial metabolisms. But just because we saw all of these compelling chemical signatures in the data doesn't mean that we have a potential biosignature. We need to analyze that data and what it could mean. So how would you rate how strong this, what is your, and obviously you need to be careful, and I don't want to have you speak for an entire group of scientists, but how strong do you think this evidence is, or is it just a first step and we need to do a lot more analysis?
Yeah, maybe I'll take your lead on that and just say I'm, not speaking for the team,
Yes.
speaking just for myself.
Yeah.
I think it's reasonably compelling.
Yeah.
I don't think it's a slam dunk. I don't want to jump the gun a little bit, and we've been misled before in big ways that have been, um, at least in the short term, kind of damaging to the field.
Yeah.
So I don't want to uh, give the public too much excitement and then let them down. But I will say this is a really intriguing set of observations that the direction that I would be leaning towards is biotic, but we need to learn a lot more about these rocks.
And how do we make that judgment in science? I came across this scale. I haven't seen it before, but I'm absolutely sure you will have, which is this cold scale. What does this cold scale measure, and where potentially are we on this scale at the moment with these potential biosignatures on Mars?
Yeah, this is one of the more well-known sort of theoretical frameworks for where you put a confidence of life detection scale. So yeah, we're thinking about how confident are we in this potential biosignature? Are we sure that it's life? That would be up towards the top.
Yeah.
Are we less sure?
That's
Based
doubtful.
on this scale, yeah, I think I'd probably put us somewhere between a 1 and a 2.
Right.
Even contamination with the rover, I mean, that of course couldn't produce these features that we saw, but there's a whole lot of steps between 1 and being positive
Yeah.
that we found something that was produced by ancient life. So I'd put us pretty low on the scale, just towards 1 or 2.
So you mentioned,
Um,
yeah, contamination. Maybe there's not too much contamination from the rover, but demonstration or prediction of biological production of signal in the environment of detection. You mentioned there's still a lot of work to do with understanding what Mars was like into the past. Have we ruled out all plausible different ways of making this the abiotic sources? Not as yet, which goes
Not
hand in hand
quite.
with
Yeah.
understanding Mars. So we've probably got a little bit of work to do, at least on 3 and 4, which seems sensible to put as 1 or 2, like you have, and then above we've got independent signals, we've got ruling out alternate hypotheses, the kind of bigger steps towards the end. So 1 to 2 seems very sensible at the moment. Lots of work to do.
Yeah, and it's really fun seeing how these quite theoretical frameworks for how we think about the detection of life that were sort of as yet really unprobed, untested maybe 20 years ago. Now all of a sudden, you've mentioned a few examples, phosphine on Venus, for example, where we're really thinking about this in a more concrete way. When can we be sure that we found life? It's exciting to live in a time where there are
Where you actually have to
many.
stress test these frameworks rather than them just being
Yes,
something on a piece of paper that doesn't have to be kind of taken too seriously.
exactly.
Amazing. It really is a fantastic time. So that brings us to the obvious question that follows. How are we going to get further clarification on this matter? So there are more samples, and presumably there have been more scans made on Mars. I don't know if they will give us any more information about this particular question or this particular rock. The data has been made available for further analysis by teams around the world. So I guess other experts are going to look at this, potentially find any issues, or they're going to support this. And they're also going to think about other abiotic mechanisms. I guess there's going to be lab work done to see if you can recreate these signals in a potential proto-Mars environment, if we get more information about what that environment might have looked like. How are we going to, what are the main steps towards getting us towards understanding whether this is biological processes operating on Mars?
We had a very good image switch over that I just saw. So you mentioned lab work. That's certainly one way. Of course, this data has been made available, open source. So people in labs around the world that want to try to test these ideas can do so.
Yeah.
Certainly, it's possible that other really clever groups of scientists come up with a way to create these features that doesn't involve life, or maybe doesn't involve as high temperatures as we originally thought. That's certainly plausible.
Maybe
And
there's
that,
some
that
catalyst on the Martian surface that we haven't thought about, or whatever it might be.
certainly possible. And of course, we welcome that kind of scrutiny. That's what science is all about. Ultimately, in terms of getting a slam dunk, requires sample return.
Yeah.
Because A few reasons. Probably the main way that you could really definitively say this is caused by life is isotopes.
Right.
Life loves the light isotopes. So if the lighter isotopes of sulfur are seen disproportionate to the heavier isotopes, that's a really good sign.
Is that a diffusion
If
issue because they diffuse faster and faster processes of
yes,
life? Okay.
from what I understand,
Because I know for
and the same
carbon,
is true of carbon.
yeah, for carbon, it's definitely you have to do the corrections. For example, in carbon dating, for example, you have to think about the ratios that life is going to take on board. Okay, so if we could get these back, we could look at the carbon ratios and
Mhm,
see if they're the ones that life would potentially favor.
mhm. Uh, Another thing is just what organic molecules do we actually see
Mm.
present in the sample? Are they long chains of organic molecules? Do we see things like lipids, fatty acids? In the organic molecules that we do see, is there chirality? I guess that is probably the big thing,
Right.
right? If the organic molecules that we see present in the sample really prefer left or right-handedness, that would tell us that it's probably related to life.
Mm.
Whereas if it's a mishmash of different chirality, then there's probably something else going on there.
So this G
Um,
band detection, it tells us that there's those carbon-carbon double bonds, but it could just be, I don't know, some alkene or something or
mhm,
whatever it might be, and it might be a very simple molecule, but
mhm,
potentially it's part of a benzene ring or something more complicated. But we
mhm.
can't tell, we can't differentiate them with just this G band detection. It's just one
Yes,
particular bond type, right?
yes. But this is all, of course, observations that would be reasonably routine in a laboratory on Earth.
Mm.
Of course, the circumstances would be tremendously exciting, but that's all to say it's not like we need to develop new types of laboratory techniques in order to get this data. We just need the samples.
Yeah.
Um, yeah.
So this is a limitation of essentially what you can pack onto a rover or do in situ with a rover from, millions of miles away, right? You can't have this entire lab doing all of these tests. We can only do so much with this rover in situ.
Yep. You need to make choices. You don't know exactly what you're going to find.
Yeah.
For example, the Curiosity rover, which is in many ways a sibling of the Perseverance rover, has a gas chromatograph mass spectrometer, SAM, which can actually differentiate between different types of organics that it finds.
Yeah.
We don't have that spectrometer, in part because we also have a complex sampling mechanism with the arm, because the idea is this was the first leg of Mars sample return.
Yeah.
That was the sort of genesis of the Perseverance rover mission. We're going to take a whole bunch of drill cores, we're going to store them so that one day in the future we can return them and do a whole bunch of analyses that we never could possibly hope to do with a robot on Mars. That's the idea.
What is, and I don't want to get into kind of a difficult political moment, but it's very important to discuss. What is the current status with respect to Mars sample return? Because obviously we've been hearing a lot about funding cuts for science over in the US. Some people I've seen talking on social media potentially saying that these results are very timely as a reminder of the importance of the Mars sample return mission. What is the status of that mission? Because it seems that if we want a definitive answer to this question, we really need to get those samples back. It is absolutely critical. So where are we with that at the moment?
From what I understand, the entire Mars sample return architecture is in flux.
Okay.
So there was a time maybe 5 years ago when I could probably quote to you a timeline
Yes.
and give you a specific architecture. Here is the sample retriever lander. Exactly, this image. Yeah, the sample retriever lander, and it's going to launch off the surface. And there's the European orbiter, which is going to pick up the samples.
Yeah.
I can't really quote that architecture to you anymore because this is all under flux,
Yeah.
all being reassessed from a cost perspective, from a timeline perspective. I think all we can say now is 2030 is probably not a realistic
Right.
timeline for when these samples are coming back. Um, we're probably looking more into the mid 2030s.
Okay.
But that, again, speaking for myself, speaking as someone who doesn't have a whole lot of inside information, um, I think the science team has done a good job of demonstrating that these samples, particularly the Sapphire Canyon sample, are really worthy
Yeah.
of meeting that high bar of return.
Yeah.
Um, and next, we just need to see what the engineers can come up with.
I as
Right.
someone who works in or worked in particle physics, you know, this feels like trying to pitch the next collider, right? We have all this amazing science, it's such an obvious use case, but it also then rams into political reality where you need to get the money and be able to do this. To us, I mean, this seems like an absolute no-brainer. Get these samples back. Let's work out the answer to this question. And it's sad that we seem to be, like I say, running up against that political reality. But hopefully, fingers crossed, those samples will come back sooner, exactly, rather than later. So let's leave that. I don't want to finish on a sad note. So if we let's imagine we do get these samples back in a timely fashion, what would these results, if they were to hold up, as we have seen? So we get these samples back, the chirality checks out, we see these isotope ratios. It looks like this really is the sulfide or iron reduction processes with microbes that we see on Earth operating on Mars. That would be groundbreaking. It would tell us that life existed on Mars in the past, which would be amazing for a start. What else would that tell us about the development of life on Mars and potentially more broadly in our solar system if those results were to hold up?
It would be incredible. Yeah, if we were able to confirm these results in the lab and we were sure that this was formed by bacteria on ancient Mars, the implications would be absolutely massive. The first thing that comes to mind is um, I would be very curious if we could if this life has DNA, right? Can it be linked somehow genetically to life on Earth?
Hmm.
If that's the case, then what we could be looking at is um,
Panspermia,
this idea
maybe.
of panspermia,
Yeah.
right? Yeah, perhaps the life on Earth and Mars had a shared origin.
Hmm.
Perhaps they both came from the same place, both came from the same asteroid,
Yeah.
or perhaps an asteroid which once long ago hit the Earth distributed life to Mars.
Hmm.
If that's
Or vice
not the case,
versa, I guess.
or vice versa,
Yeah.
yeah, maybe we're Martians originally. If that's not the case, I think that's an even more exciting possibility. If we can somehow demonstrate definitively that this was a completely separate
Hmm.
Genesis moment,
Hmm.
uh,
That's two planets
yes,
next to one another, both of which had life. That kind of would
yeah,
seem naively to skyrocket the possibility that life is everywhere,
yes.
right? It's more likely
Uh,
than not.
Suddenly, I mean, there's other possibilities for distinct Genesis moments looking out into the solar system
Mm,
that are also,
mm,
Europa,
yeah,
Enceladus, Titan, etc. So suddenly we could be looking at a universe completely chock full of life. And of course, we still don't know how intelligent life
mm,
develops, and that's a big part of uh, the Fermi paradox.
yeah,
And well,
yeah,
maybe intelligent life is the thing that's rare,
yeah.
but it would still be absolutely monumentous if we could demonstrate that there's a second Genesis moment on Mars, and it would. I would, I was very biased in my position. I feel like that alters the course of human history,
Yeah,
and that alters the way we think about the universe in a really dramatic way.
absolutely. In terms of the context of these rocks as well, I think you mentioned that they're mudstones. They were laid down. They're close to the surface, which potentially suggests, and this is me doing amateur geologist, that they're relatively young, right? If they're at the surface, so could this suggest that Mars was potentially habitable um, less far back into the past than maybe our standard timelines suggest?
Certainly possible. I think there's still a little bit of a debate among the team as to whether these sediments that we found in this river channel are older or younger than the igneous rocks that we find just to the north or south.
Right.
But I think most people are convinced that they're probably younger. If they are younger, yeah, the possibility that you mentioned, they could be significantly younger.
Mm.
They could be half a billion years
Mm.
younger than the material surrounding it, in which case that extends the range, as you suggest, of the habitability of Mars by hundreds of millions of years. That's certainly a possibility. We don't know the relative timing of all of these things, though. Even if it is younger, it could be 5 million years younger,
Yeah.
and it's still 3.9 billion years old, for example. We really don't know. That's why I think all of the samples together are really compelling, even if they're not directly tied to the story of life. Rather than the really coarse crater surface age dating, we can directly get the crystallization age of the igneous minerals in the samples and tell you exactly how old it is. And that's a really important part of the story too.
So the bottom line is we have to get these samples back to Earth. Is that fine as a signing off point?
You're saying it, I'm not. No, but I think they're a very compelling set of samples. I think if we want to answer these big picture questions about the potential biosignature, potential history for life on Mars, we will need the samples to come back. So,
Absolutely fantastic. I think that's a brilliant place to leave it. Henry, thank you so much for going through all of those questions with me. I think people are really going to enjoy listening to that. I will make sure that all of your work, your links. I know you do some writing as well about these topics. Do you want to point people to anywhere where they can find out more about this work and about the things that you write?
sure, a little bit. I help run the Perseverance Rover blog. So if you just Google Perseverance Rover science updates, every week we give an update on what the rover's doing, which can be kind of fun. Usually, if you look at a map of where is the rover now,
Yes.
you can sometimes see us just driving in squiggles, and you might be wondering, what are we doing? If you look on the blog, often it's me posting, often it's other people posting. We explain why we're doing what we're doing, the types of investigations we're currently doing with the rover, and why we think they're worth the rover's time. In terms of my other socials, I don't think I have many others. I guess I'm still on Twitter, Henry_Manelski, and I tweet about science occasionally. But I think that's all I want to plug.
Yeah.
Unless anyone listening wants to hire a postdoc, then please reach out, especially if you're based in Europe. But I think that's all I have to add.
Absolutely brilliant. Henry, thank you so much again. I really, really appreciate you taking the time. I've learnt an awful lot. Fascinating stuff. And hopefully the next time we find out more about these samples, or we have another interesting detection on Mars, we can get back together and discuss it again, buddy.
Yeah, I'd love to talk about it more. Thank you so much for having me on.
It's been a great pleasure. Thank you very much, Henry. And I'll talk to you again soon.
Talk to you soon.
Take care, buddy. Take care. Bye now. I want to know what you think, because you're the scholars of enlightenment that I do this for. So please take a moment, if you wish, to let me know down in the comment section. And if you like this video, please consider leaving a like, subscribing, setting up notifications, and sharing this video more widely. I can't tell you how much these simple actions help me out and how much I'd appreciate it. Thanks for watching. Thanks for being scientific. Thanks for being bad.